Biological samples extracted from the crime scene usually contain very small quantities of the DNA, like human samples or bacterial samples. These samples need to be amplified for further analysis to be performed. To amplify a DNA sample extracted from the crime scene, a method called polymerase chain reaction, PCR for short, is used. This tool and another tool called restriction endonucleases, which is the subject of our next topic, are crucial tools of molecular biology and are incorporated into a number of strain typing technologies. These terms might sound intimidating, but practically the application of these tools is simple and easy to understand. I want you to focus and enjoy the new set of knowledge to build on what you've already learned. PCR has transformed the way that almost all studies requiring the manipulation of DNA fragments may be performed because of its simplicity and usefulness. PCR is a rapid, inexpensive, and simple way of copying specific DNA fragments from minute quantities of source DNA material, even when that source DNA is of relatively poor quality. For example, biological samples extracted from a crime scene that might be contaminated or degraded. One important reason of why you may want to amplify DNA is to simply create multiple copies of a portion of DNA which is very rare. For example, a forensic scientist may need to amplify a small fragment of DNA from a crime scene. There are two major components of any PCR reactions: the polymerase enzyme and the primers. A polymerase will synthesize a complementary sequence of bases to any single strand of DNA. This is very useful because you can choose which gene you wish the polymerase to amplify in a mixed DNA sample by adding a small piece of DNA complementary to your gene of interest. These small pieces of DNA are known as primers, because they prime the DNA sample ready for the polymerase to bind and begin copying the gene of interest. During a PCR, changes in temperature are used to control the activity of the polymerase and the binding of primers. To begin the reaction, the temperature is raised to 95 degrees Celsius. At this temperature, all double-stranded DNA is melted into single strands. The temperature is then lowered to 50 degrees Celsius. This allows the primers to bind to the gene of interest. Thus, the polymerase has somewhere to bind and can begin copying the DNA strand. The optimal temperature for the polymerase to operate is 72 degrees Celsius. So, at this point, the temperature is sometimes raised to 72 degrees to allow the enzyme to work faster. There are now twice as many copies of the gene of interest as when you started. The cycle of changing temperatures, 95, 50, and 72 degrees, is then repeated, and 2 copies become 4. Another cycle, and 4 become 8, up to 30 to 35 cycles. After amplifying the gene into many millions of copies, it's possible to run the amplified DNA out on an agarose gel to visualize it. A bigger visible band contains more copies of the gene of interest. The resultant DNA fragments are then detected through a process called gel electrophoresis, which you will learn in later topics. Okay, then if you have a small body sample or bacterial sample, you know that you have to multiply the region of interest using PCR. Next, another important technique for the diagnosis of microbial samples is discussed. To summarize this topic, PCR is used to amplify a given region of DNA. Polymerase is used in PCR to synthesize nucleic acids. A primer serves as the starting point for DNA synthesis. Finally, DNA fragments are viewed by gel electrophoresis. One point I want you to know: polymerase is a bacterial enzyme. Well, I'm not gonna give you any more details. I will leave this for your discussion forum to discuss.